hello to everyone. Please let me know in the chat box if my voice is loud and clear. Okay, thank you. That's great. Thank you for the confirmation, everyone. I'm Dr. Tanushree, Senior Faculty associated with Biotechnica. I welcome you all to the exclusive webinar on Computational Chemistry and Materials for Pharmaceutical Therapeutics, brought to you by Biotechnica in association with Altum Technologies. Today, we have with us proficient personalities in their respective fields. Mrs. Divya Shanmugarajan, Senior Application Scientist, Life Sciences, Altum Technologies. Dr. Anand Krishnamurthy, Senior Manager, Industry Process Consultant, Biovia Dassault Systems. Dr. Krishnamurthy is here to enlighten us on Generative Therapeutics Design. Dr. Mark Munier, Principal Application Scientist and Science Council Fellow at DS Biovia. Dr. Munier is here to enlighten us on Computational Pharmaceutical Materials Science. We'll be starting this session with the insights from Mrs. Divya, then Dr. Krishnamurthy, followed by session from Dr. Munier. Also, there will be panel discussion as well as Q&A session at the end. So without taking much time, I welcome Mrs. Divya on board.
Thank you, Tanushree. I'm sharing my screen. So, it's great to meet everyone in this virtual platform, and today is going to be very interesting, I could say that, because we are going to explore how the computational chemistry and the materials for pharmaceutical therapeutics would give a solution in various domains. I am Divya here from Altum Technologies, Senior Application Scientist for Life Sciences, going to present and going to moderate the session. So before wasting time, I'm just going directly to my company and taking great privilege to introduce my company. Altum Technologies is a company we started in the year 2011, and for past more than a decade, we are a leading design and a manufacturing innovation in India to the various products and services. We have headquartered in Bangalore and had a presence all across the country. We have more than 400 plus customers in various industries such as aerospace, automotive, healthcare, consumer goods, life sciences, and we are a platinum partner for Dassault Systems and the statuses for past 10 years. We are also into the 3D innovation platform company. We believe that we want to provide end-to-end solution from a concept stage. We are a team of designers to the final stage. We are the manufacturer, and we do so using Dassault System tools and software. And here you can see our solutions and prestigious awards. And these are our customers' question list. And we're also being facilitated a lot of awards and journeys from our government awards, access to Dassault Systems partner, many more. With this, I will be very privileged to introduce my company. Now, moving directly, I'm going to introduce a small words about Biovia. Biovia is a leading software and a provider for chemistry, materials, and biosciences research for pharmaceutical and biotechnology industry, headquartered in United States. It's represented in Europe and Asia. Biovia sophisticated simulations, modeling, and quality management systems enable customers to discover, design, and provide innovations products globally. Biovia also provides solutions to more than 2,000 plus companies in life sciences, chemistry, oils and gas, consumer packaged goods, and deliverables to academic and government entities. So, such a huge company and joined with Altum Technologies, I am proud to announce this. And with this one, I would like to introduce my senior and as well as eminent speakers, Dr. Mark Munier and as well as Dr. Anand Krishnamurthy. Welcome you all from Altum Technologies for this wonderful session. Without waiting, I am handovering the session to Dr. Anand Krishnamurthy, sir. And before that, I would like to introduce few words about him. Dr. Anand Krishnamurthy, who is received his PhD from University of Madras, and he is in Dassault Systems for more than 20 years, and he is having a very wide experience in the life sciences for more than 23 years. Moreover, he has published various papers and reputed person for a Biovia Discovery Studio, and he has a solutions. There are lots of works in various domains in simulations, modeling, antibody modeling. So like this, I can pile up its discoveries and its solutions for various academic as well as for industry solutions. With this, the short introduction, I hand over the session to Dr. Anand Krishnamurthy, sir. Please kindly take up the session.
Thank you very much, Divya, for giving such a nice introduction. Let me just share my screen to start with the presentation part, and let me know if you're able to see my screen. Fine, you're able to see my screen? Divya, if you can confirm, please.
Yes, sir, it's visible.
Okay, thank you very much. Okay, with a nice introduction from Dimya, and thank you very much Biotechnica and team Altem to arrange for this wonderful meeting, basically, or discussion based meeting, or a webinar, or a conference respectively, to give and introduce two specific aspects, how we are going to bring in both life sciences as well as material sciences into the pharmaceutical industry to have the best of the innovation and discovery is what we are going to look at. And in my presentation today, I'm going to talk about one specific aspect or one specific area that's called generative therapeutics design. So what is this generative therapeutics design? We do some generation basically of designing some therapeutic molecules. That's what we look at. And we will go through some of the specific challenges that is addressed in this particular sector, and how these challenges is addressed by Biovia's Dassault Systems, and what are the key benefits and value that adds to the pharmaceutical domain in terms of innovation and discovery is what we will be looking at. So typically, what are the challenges in pharma? Every pharmaceutical company want to come out and innovate one molecule. It could be a drug molecule, small molecule, it could be a monoclonal antibody, it could be a peptide inhibitor, whatever it is. So it's going to be ultimately a molecule that they want to come out, and it could be a molecule that could be designed for a specific purpose. It could be a molecule that could be searched or screened across a set of databases. So what is the challenge here? The challenge is we have millions of molecules that are available virtually in the databases. How do we come out with one specific drug molecule, or let us say few specific drug molecule that basically takes care of different properties? That is totally patentable, it's manufacturable, it's stable, it's potent, it has some desired properties corresponding to that particular disease condition that we are working on. That's the most difficult task. And most of the time, to identify, understand, design this molecule, it takes a huge amount of time, something like 15 to 16 years basically for the whole discovery cycle, and coming out with the drug into the market, and an enormous spend or expenditure on the money, which is around the tune of around $2.6 billion. And that's totally huge. And what we do achieve, pharma companies doing that, they want to be the first to patent for that particular disease condition, a drug molecule, so that they capture the whole market, or in addition, they be the first patented organization related to that particular disease. So this is the fight, this is the challenge, and these are the different areas that needs to be addressed. And how do we address it? What are the different ways by which we can address it? So this is one of the CEO's letter from Novartis. It's called Vas Narasimhan. Everybody would know about him, definitely. And the aspiration and strategy of their growth basically was to become an industry leader using some advanced technologies and new technologies basically, and reduce the time from discovery to commercial which is typically now 10 to 15 years. But we have seen in this COVID pandemic how we came out with a vaccine, how we repurposed different drugs, came out with vaccine within a period of 18 months, basically. That's the level of acceleration that we had. And there was a big contribution from the computational discovery part of it. But although there was a big contribution in the computational discovery part of it, there are different aspects that is attached to it. Basically, how do we accelerate it? How do we see the efficacy? How we see the specificity of that molecule is that we have to address. So with the CEO's letter in terms of being setting up a strategic objective to become an industry leader in this sector because of the competition and the need to come out with an innovative new molecule respectively, Dassault System has a solution, and the video goes through that. You can listen to that video, I suppose.
5 years, thousands of molecules, many iterations, frustrating restarts just to produce one lead candidate. AI and machine learning can help you explore drug candidates faster to meet a specific target product profile. Generative therapeutics design of Dassault Systems gives you tailor-made compounds with the best chance of success. Combining the creativity and knowledge of medicinal chemists with powerful machine learning tools and cloud computing results in better outcomes, faster drug discovery. Generative therapeutics design transforms discovery. Gaining a competitive advantage with AI is the future. It's now.
So what you have basically seen in this video is basically identifying a target product profile. So for a particular target or a set of target, I need to identify a lead compound that actually matches to certain set of profiled properties. That's the idea behind that. How do I do it using an artificial intelligence based approach or a machine learning based approach and do an enumeration based on that? That's the whole thing about this particular product, which is very intuitive, very user friendly, and which gives you a discovery in a very short interval of time. I'll show you some of the advantages and some of the value that this particular product or this particular generative therapeutics design has to offer. So what is the bottleneck in this particular discovery, what we see? And the strategic objectives basically with respect to what we saw in Novartis is totally aligned to this. So it takes 5 years. That's an average time that is spent in discovery. And when it is discovery, it is not going to be one molecule that comes out from an pharma organization, innovative organization, but it can be anything between 2,000 to 4,000 compounds to find a viable candidate. So synthesizing 2,000 compounds or synthesizing 4,000 compounds is going to add up on an enormous cost, as well as going to be a lot of time that people have to spend, or the synthetic chemist and the medicinal chemist have to spend. And this is the flow that has been followed over the decades and decades. And now, with these generative therapeutics design and artificial intelligence in place, it has been a different game altogether. I'll show you what are the different games associated with that, what is the different approach associated with that. So I choose certain set of target properties. I have to have a compound which is highly potent. I have to have a compound whose selectivity is off target, which has a blood-brain barrier penetration, which is not hepatotoxic, which has good solubility, and many other properties. So when I take a single compound or a multiple set of compounds, I have to iteratively develop a lot of compounds, basically, which matches to each of these target properties. So the iterations is done cycle on cycle, to generate millions and millions of compounds, which starts with few compounds respectively. So when you see at the end of the 4th iteration or the 5th iteration respectively, I have a compound which matches to 4 of my target properties, and that could be one of the ideal candidate for my disease condition or a drug target that I am focusing on. It is not only limited to these 4 target properties, but can spread across multiple other properties which could be included, like metabolic stability, the developability, the ability to get patented, ability to get synthesized, synthesizability, and so on. So you see that I create a target product profile which is based on multi-objective optimization. So I'm optimizing a set of multiple objectives over here. And when I do this, basically, I come out with a compound. So this is what is not being done in decades together. So the process is usually trial and error. The target properties usually is incompatible and it's usually competing with one another. You need to do physical experiments for each of them. It's too long, too costly. And many other critical properties should be considered when you want to use, when you actually cause a drug candidate and it fails in the downstream process. So creating a target product profile using a multi-objective optimization is the solution to this. And this solution is offered by Biovia's what is called Generative Therapeutics design. The first and foremost part of it is basically to generate a set of compounds using repetitive iterations and testing them with computational approaches. That is purely virtual. So you have a few set of compounds, you generate multiple thousands and millions of these compounds, test against a particular target, score them, and then identify those compounds which are useful and remove all the other compounds basically. So you come out with certain set of compounds. The medicinal chemist looks at these compounds and sees the ability to synthesize these compounds and also looks at some of the functional aspects of these compounds which really work on being an agonist for a particular protein target or acting against a particular target to combat a specific disease condition. Then it goes back to the virtual part where an enumeration is done on that particular compound and you get a series of compounds that is available and those compounds would go on to become an ideal candidate for that disease conditions. So who are the people involved in this? It's not one set of people which we are targeting. We are targeting people from head of research, from molecular modelers to data scientists who have a capability to use this generative therapeutics design. So why generative therapeutics design? The reason for that is drug design requires a rich set of capabilities, and the rich set of capabilities is not limited to some functional properties. It is open to many ways by which you generate molecules, how do you select molecules and go on to do the predictions. With Biovia's experience of 25 years in this scientific track record, we have a capability to generate compounds, select specific compounds based on its functional capabilities and properties, and go on to predict some of the properties that is key for this target product profile that we are doing. So what does the generative therapeutics design solution offer from Biovia? So what you do, you basically select a target product profile, select from Biovia's machine learning models that you have. So I want a molecule that passes through the BBB, that passes through the CNS system, that has least toxicity of mutagenicity and the least toxicity of carcinogenicity respectively. Using that, I generate a set of molecules and iteratively the program starts generating millions of molecules respectively. And each molecule is mapped against each of these targets to identify which of these molecules passes satisfactorily over these specific properties and is virtually examined from time to time. And also any structural transformation that occurs within these molecules is also identified. And a complete model is built based on these specific compounds with this target profile. That's the idea behind generative therapeutics design solution that Biovia offers. So who has gained from this? So this is one of the examples where we have a company in the United States. It's a pharmaceutical company. They have used generative therapeutics design to identify a set of compounds that meet all the target properties. And chemists use these GTD as an ideation engine to show the trade-off for improving the molecular properties and also helped chemists to be more imaginative about the molecules that they want to synthesize and to get less misses on the activity part of it. So what does accelerating drug discovery success factors come in? So basically you need a human in the loop type of learning. You have an AI in the loop for the learning to suggest to the medicinal chemist at the lab, basically lab in the loop learning, where you will be able to synthesize only the required molecules rather than not all the molecules that you screen. That's the idea behind the whole accelerating drug discovery process using generative therapeutics design. The benefits, the value, if you see, the traditional drug discovery approximately takes 5 years to discover a compound, which actually spans across 4,000 compounds which are synthesized. And AI-based drug discovery approximately is 3 years, and which takes around 1,000 compounds that needs to be synthesized. It should be much less than that. I will give you some of the examples as we go on in the discussions following this. So the estimated savings per optimized lead may be across from 8 million to 12 million respectively. So with this solution, Biovia's generative therapeutics design and a strong scientific track record with Biovia for over 25 years respectively, offering a cloud platform with minimum IT infrastructure, an industry solution that has a very intuitive and easy user interface, ability to integrate to multiple machine learning models and algorithms, and also support industry collaboration between the researchers, the synthetic chemists, medicinal chemists, and across the regulatory part of the any pharmaceutical organizations respectively. These are the key strengths that Biovia offers in its generative therapeutics design. So that's my presentation, and thank you very much for listening to the presentation. Thanks.
Thank you so much, sir, for your wonderful presentation. And thank you so much, sir, for your wonderful presentation. It is really great, and people who is working on a pharmaceutical industry will really get benefit out of this technology. So with this, I'm just feel very proud to introduce another eminent scientist and personality from Biovia, Dr. Mark Mueller. Dr. Mark Mueller, he works as a principal application scientist and Science Council Fellow at DS Biovia, and he holds his master's from Pure and receives his doctorate in chemistry from Marie Curie University in France. And he joined Biovia in 2000 as a product specialist for materials modeling. Mueller's research interest includes a study of nanodielectrics, the simulation of polymeric materials used in membrane technology, pharmaceutical material sciences, and which is a most upcoming and a growing field for all the peoples to make it for an accelerating a drug discovery process. And material informatics is going to play a huge role. With this, I welcome Dr. Mark Mueller to express and express more about how the materials could helpful for people who is working in a pharmaceutical industry. Hand over the session to Dr. Mark Mueller. Please, sir.
Hello, good morning. Thanks for having me. Thanks to Altem and Biotechnica for inviting me. I'm going to try to share
Yes.
the screen. No. Okay.
Your screen is this.
You should see my screen in full now. Yeah, can you confirm, please?
Yes, sir. Your screen is visible.
All right, great. Thank you very much. Yes, so today I want to talk about computational pharmaceutical material science, and which basically relates to the use of common material science technologies applied to the development of pharmaceutical therapeutics. I am work for DS Biovia. I'm based in Cambridge in the UK. And so Anat just presented us the top left part of the life cycle development of a drug. He typically mentioned about 5 years from target discovery to lead ID. And the entire product development can take up to 14, 15 years, depending. So after you have the active compound from your discovery process, what you have to do basically is just to transform it into a product. right? You have to go from the active molecule to, say, a tablet, a pill, or anything. So you have to wrap it up, and that really becomes just a problem of chemistry and physics. Really, it's a productization of the active. So we are back to very similar to making a shampoo, a toothpaste, or anything. So what do we do in those 3, 5, 4 years of development, pharmaceutical development? We try to characterize the active. We're trying to see, to predict its crystal shape, its morphology, and very importantly, we try to assess its solubility and the stability. So solubility, excuse me, because that is very important for the bioavailability of the drug in the human body, and stability because you have to package this API with excipient, and it has to stay together. So we really have to go through the process of characterizing as much as best as we can and as early as we can the materials in order to avoid any issues that could arise later down in development that could be expensive to remedy. Say a polymorph, some of those drugs can have polymorphs, and if you discover a second polymorphic phase later in development, that can be very costly. There's another tool we won't mention today about Cosmotherm, which is about computing solubility, and I say a few words later. So why do we, so that is the background, and so we have a drug from the discovery process, and we have to characterize it. So we go in the lab and we do NMR, X-rays, and so forth. But why use in silico method? Well, the aim, as I say, is a risk assessment, is to try to predict any potential issues that the drug might have later in development, such as low solubility, polymorph, a need for co-crystal, and so forth. You might have very limited API because this is an early stage of development. The complete organic synthetic route is not fully established yet. So you might have to, you don't have much yet. And so you also want to screen the solid form. So you will work with different salt. You very often will create a salt to increase solubility. And for that, you might need to get the crystal structure. And that we can do with in silico too. We can also look at polymorphism, morphology, and so forth. So the aim really is trying to reduce a trial and error experiments and try to predict those physical characteristics of the active. So the tools that DS Biovia provide to that end is called Material Studio. It's a molecular modeling package which contains so many different methods that can be applied to the problem of interest here. For example, predicting the crystal structure, or look at polymorphic forms. So that is a different crystal form of the same compound. And the morphology is relevant also for predicting crystal shape and sizes. We will look also at solubility and stability, that are the two main physical characteristics of interest in this development process. We want to enhance solubility to increase bioavailability and bioefficiency, and we want to increase stability to have a product that have a long shelf lifetime. So the first thing we do with our in silico tools is to look at the crystal. So we have a Material Studio visualizer where you can look at the crystal structure, and you can measure all sort of geometric values, the angles, the distance between the compound. Is there any void? Could you form a hydrate? Because those blue things here show that there is a bit of space in between the drug molecules. You can simulate an X-ray. You can upload a CIF file from the CCDC database and then simulate an X-ray and see how that compares to the one you currently have. So the first tool that the computational chemist will do in this development process is often the assessment of the crystal structure. right? So we start with a 2D or 3D excuse me, representation of the drug. We have an X-ray, and then we try to guess the crystal structure. That is not always an easy game, but we have the tools to automate this process. You can also use simulation of the NMR. So here we have a tool called CASTEP, NMR CASTEP, which allow for the predicting the key magnetic resonance properties of the molecule and the solid state materials. So basically, you will get the chemical shift tensor of the compounds, and that will allow you to refine better in particular, the hydrogen atoms that do not appear in the X-ray, they're too light. So this method is very much used, again, in the large pharma company to refine the crystal structure of the drug. Polymorphism is also something that one is concerned when it comes to drug development, because, as I say, like in the case of Norvea, that this polymorph that appeared very late, or even after the drug was released, it's very costly then to go back and redevelop a new form. So how do we do that? So we take the 2D, we take the diagram of the drug, and we create as many crystal structure as we can, and using some clever tricks and computing the molecular interactions between the compounds in the crystal cell, we can assess the stability and the likelihood of the active ingredient. So we typically plot density versus the lattice energies at the stability of the drug, of the crystal cell, if you wish. And that's cohesion. And then if we have too many points down here, then we know that this drug is likely to present different polymorphic forms. We can also, what you do in the lab is you do series of X-rays at different solvent, at different concentrations, and you look also how they cluster together, right? And so we can also use different tools like quantitative phase analysis in order to identify all those different forms. An important aspect also is that most of the drugs are given to us in the shape of a tablet, a pill, a tablet. And I would say just that I gave a presentation just a few weeks ago at the 2021 Biovia conference on this very topic of multi-scale simulation of the tableting process, shown here. So when you mix the drug, the API, with the various excipients, and then you try to reduce particle sizes, so you mill and granulate the product. You dry it, compress it, and then compress it, and then get a tablet. So the particular size and shape, of course, are very important in this case, and that, as I mentioned, can be predicted thanks to the morphology predictions. The compressibility of the API will also be relevant here. So here we investigate mechanical properties. That's what we can predict with our tools, such as the CASTEP one I mentioned before. There are other methods. We can look at the stability, degradations, class transitions. And then a very important point in the development of the tablet, a common issue is punch sticking. That's what happened when, here in number 2 or 3, you have some of the material that stick to the punch that is used to compress and make the tablet. So, yeah, so that punch sticking is sometimes an issue. And so we can investigate the interaction between the active ingredient and the excipients that are here, the binder, lubricant, and so forth, and how these interact with the surface, the steel mostly stainless steel surface of the punch. So I mentioned we can predict the crystal habit of the crystal, so that is the tool of Material Studio called MS Morphology, and which goes from the crystal cell and tell us in different conditions if we're going to have a needle, which we want to avoid because they're usually brittle and they're not great to make a tablet of, and we try to have something a bit more isomorphic. So we can calculate the most important crystal phases of the drug crystal thanks to this software. Regarding stability, this is a study we've done a few years back with Sanofi, and they wanted to develop an in silico risk assessment of API stability with respect to the auto-oxidation, that is the hydrogen abstraction. So that's when you see in the top right corner here, when your compound loses the proton, and there is an abstraction of a hydrogen, and that has consequence on the stability, of course, of the compound. And this happened due to interaction with the excipient in which it is mixed. So what we did is to take a series of known compounds and which experimental data was known, and then we computed the hydrogen abstraction of that is the molecule H and the molecule without the extra hydrogen, and then calculated the energy difference using quantum mechanics here, using the density functional theories. And we've done that with various different settings, because there is a trade-off between the accuracy and the length of the computational calculation and the accuracy that you want. So we found a series of settings, and now the chemists, and you can see again a presentation from Sanofi at the Biovia UGM showing you the compound toolbox. And the compound toolbox is like a tool, a web page, where in this case, the chemist can upload a compound API and then ask for the likeliness of being subject to hydrogen abstraction. We can also, for the stability, an important characteristic is the glass transition temperature of the drug in amorphous state here. And so, because sometimes yes, the drugs are made in the amorphous state rather than crystalline. And so in this case, what we really want to assess is this glass transition. But we also want to see the effect of water, say, water humidity, that can have on the glass transition, usually to reduce TG. And this you can just do using the software. You can mix the API and say the polymers in which it is dispersed, and then run some molecular dynamic simulations at various temperature points and check how the density varies. And at the point where change of slope of density temperature varies, then you can have the TG as here. I mentioned the Cosmo firm, based on the tool, based on Cosmo RS methods. That's a recent acquisition of Biovia of what was called at the time Cosmologic, which is now renamed Solvation Chemistry within Biovia. And what the solvation chemistry tools allow you to is to predict the API solubility in solvents or in mixture of solvents. And that's really a key physical characteristic that the chemist needs in order to develop its drug. And it's not just in solvent, you can also do it in polymers. You can look at phase diagram, and more recently, some work was done with the tools to look also at drug delivery and interfaces. So in conclusion, I would say that using the predictive sciences in drug development allow for in silico risk assessment. We want to predict as early as we can the potential failure or issues a drug might have, such as a low stability, low solubility, different polymorph, and so forth. We need to assess the crystal structures, so we need to compute that, and we can do it with the tools of Material Studio. We try to correlate the API chemical structure with the physical properties. And we can screen many potential formulations. That's the advantage of using in silico, like Anand was showing us before, in this GTD process of screening as many compounds as we can automatically and using machine learning. Similar tools can be used here to screen many different potential formulations in order to retain the one with the optimal physical properties. So we try to avoid unnecessary trial and error experiment. At the end, we want to reduce time to market and save development costs. And thank you for your attention.
Thank you so much, Dr. Mark Mueller, for giving a wonderful presentation about how the Material Studio will have a great application in the pharmaceutical industry. And with this, I am moving to the panel discussion. Let me share my screen. So, audience, we have come to the panel discussion. The first question is to Dr. Mark Mueller. Who are the users of such solutions, such means Material Studio solutions in a pharmaceutical industry?
All right, yes, good question, thanks. So typical users are the analytical chemists that work in drug development. They can be a formulation chemist, or they can be computational chemists. They are usually in the large pharma or in biotech that focus on the optimization of the formulation. So they are basically chemists trying to predict the physical and chemical properties of the active ingredient.
Thank you so much, Dr. Mark Mueller. I am moving this question to the Dr. Anand Krishnamurthy. And nowadays, it's becoming everywhere, and we can hear the lots of talks and comments and blogs are coming on artificial intelligence and machine learning. And particularly now, I imagine a drug discovery. Don't you think this is hype today? And also, I would like to know about it, how this AI, ML, and imaging can play a vital role in drug discovery and development cycle.
Thanks, Divya, for this wonderful question. Yes, I do agree that there are a lot of buzzwords calling for artificial intelligence, machine learning, and of late, imaging, video analytics. So many things are coming in place, and of course, it has its own root and its historical presence. Let me little bit go back to historically. When we take sometime in the period the 1960s, we had the first 62 specifically. We had this first QSAR model which was developed basically by Hans equation. And this was basically to study the Hammett constant to identify the chemical reactivity or predict the chemical reactivities. And that has been a big practice. Although people started not believing much on that for 15 years after Hans equation came in. And following that, sometime in 1978-80, people started realizing the importance of it. And that was a time when Biovia introduced a program called as TopCat for Toxicity Assessment based on computer-based technology. And that was one of the premier and first programs which used machine learning, basically a Bayesian algorithm to assess the genotoxicity of the compounds. Today, regulations across pharmaceutical industry demand organizations to test for the genotoxicity, basically the mutagenicity, carcinogenicity, and toxicity of their compounds, either using a machine learning model, basically an in silico approach, or go through a experimental approach. Experimental approach is too costly. If I'm going to take mutagenicity itself, it works out to around 3.5 lakhs per sample. And if they're going if we're going to test 10 samples in a year, it is going to be huge amount of money. So it will definitely multiply as it goes on. So machine learning was one of the best approaches that could definitely help these people in the pharmaceutical industry. But machine learning does not mean that you take a set of molecule, run it, and you get your output. That is not how it is defined in a regulated environment or a regulated pharmaceutical industry. So you need to have certain guidelines that is set up. And these guidelines were set up by what is called the OECD, Organization for Economic Cooperation and Development, that is totally defined and accepted all across the world, which has 5 defined basically definitions. One is a defined endpoint, an unambiguous algorithm, a domain of applicability, a goodness of measure of it, and a robust and good predictive nature. So that was the case till artificial intelligence and machine learning came by in very heavily in 10 years back or 15 years back, I would say. What was the reason why we could not use other AI and ML based approaches was because of the amount of data that is generated. So with a huge set of data that is generated today, you can use machine learning for target identification, finding a lead, synthesis of compounds. What is compound that is synthesizable? What is not synthesizable? What is the mode of action of a compound? Whether a compound has a polypharmacological capability or a property. All of these things can actually be identified and also defined basically. So I'll give you an example before I complete the answer to this question. I'll take the example of one of the organizations called Extentia. Extentia is the first organization to come out with an AI-based drug for obsessive compulsive disorder. It's called CDI DSI 0038, if I remember correctly. And it's in the stages of clinical trials. And you won't believe it, when they bought it in clinical trials in January 2021, I think January 2020. Eventually this year they have 3 other products in the line for clinical trials. So going by, what is the advantage of that? Where they used to synthesize 2,500 leads, they ended up synthesizing only 250 leads, which means that they only use 10% for the synthesis. That's the power of artificial intelligence and machine learning in bringing a drug to the market. Not only that, as you put up the word called imaging, molecular imaging is extremely important. Today, everybody would have heard of AlphaFold 2, which is basically related to the protein folding. And imaging is a key criteria by which the folding process, protein folding process is being understood. And Google's DeepMind is exclusively working on that. There is a database available with that, and people have the full capability to Google that and get the database and the available information pertaining to that. So it's totally pronounced. And tomorrow, whatever drugs come by in the next few years will be all AI-based drugs. That is what we expect. And there is a huge boom that is going to come by. Thank you.
Thank you so much, sir, for giving us such a brief explanation by combining artificial intelligence, machine learning, and application of imaging into drug discovery process. So, moving to the next question. So, this question is to Dr. Mark Mueller. Do you need to be a simulation expert to use this software?
Simple answer is no. You don't need to be an expert. Many of the tools are packaged in a way where basically the user can simply sketch the compound and request particular properties. And what's under the hood, the quantum mechanical calculation and the force, can be automated and can be selected by by an expert. But once, like in the case of the Sanofi autoxidation that we developed with Sanofi, this was, yes, with formulators and expert in computational chemistry, but once this is well defined, when the workflow, how to compute these properties is defined, then it is made very available to the rest of the chemists that are not simulation experts, where they can just sketch their compound and click a button and say, give me the hydrogen abstraction propensity, and that's it. So we can, you need some expertise in simulation to develop the workflows for the properties that you require. But once this is done, it can be packaged, and then the rest of the non-expert scientist or chemist can then use technology.
Thank you so much for the explanation, Dr. Mark. I believe that now the audience will reach this point. We don't need to be expert into use the software, because only you are developing, you need to be expert. For end user, you should just know workflows, or you have to know what is your your questions, and that will be answered using this kinds of methods. So going back to the next panel discussion question. So this question is to Dr. Anand Krishnamurthy. So, Biovia's generative therapeutic design, I believe that is what you have presented today. And you have been told that it offers V plus R solution. How one experience this generative therapeutic design for accelerating their innovation in drug discovery?
Okay, wonderful question. A good observation of the presentation, I would definitely say that. Okay, generative therapeutics design basically combines virtual and real. So, what in the innovation life cycle basically? So, what I mean by that is a computational chemist will go on to generate multiple compounds that modulates the activity of a protein target associated with a disease condition, and a medicinal chemist synthesizes it. Assume that a computational chemist enumerates 10,000 compounds, and you don't expect the medicinal chemist to synthesize all of them. So his company will definitely throw him out or kick him out if he is going to synthesize 10,000, 20,000, or 100,000 compounds. So there should be a way by which he should have a set of compounds that needs to be synthesized and how it has to be done. So that is the basis of what is called as a virtual plus real. And when certain compounds are specifically identified which are functionally important for this particular target, an enumeration can be done circling around those compounds. And that is what a computational chemist will add on. So it is both a capability of virtual as well as a real, which gives them a winning ideal candidate for that particular disease condition or the drug target. So how does one experience it? That's a very good word. How does one experience it? It's not just a usage. I have a software, I use it, I get the result, I publish the result and get it done. How do I experience it? Biovia's generative therapeutics design is a cloud-based integrated solution that will optimize your drug discovery, design, and that will definitely help you save millions of dollars to the organization. So if I am a medicinal chemist, I'll have certain roles associated with my activity or function. If I'm a computational chemist, I'll have a low role associated with me. If I'm an assay scientist, I will have a role associated with them. Each of these role is contributed by set of apps, which forms the basis of generative therapeutics design, which is innovation based, which is collaborative in nature, which is shareable in nature, and of course, which also allows you to optimize and apply machine learning programs onto it. That's what I have on that.
Thank you so much, sir, for giving a wonderful solutions and then answer. I believe everyone understand about how the generative therapeutic design will benefit in drug discovery process. So we have a last but not least question to Dr. Anand Krishnamurthy. Can you describe the Biovia's portfolio covering the entire pharma as well as the biotech life cycles from ideations to the research to clinical trials? Please, sir.
Okay, wonderful. That clearly describes what Biovia has to offer for the pharmaceutical and the biotech sector as a whole. So definitely Mark also would definitely agree the Biovia's portfolio that I'm going to actually describe. And so Biovia's portfolio starts with what is called the collaborative sciences, basically, which is more oriented over the research community, and of course to part of scientists and other people who work on the pharmaceutical biotech sector, basically, and of course even to other industries. I would not deny that. And Discovery Studio, Material Studio, Generative Therapeutics Design is some of the specific tools which addresses these people for specifically for innovation and discovery. And we have certain set of portfolio of products pertaining to the development lab where we have a capability to have an regulated set of experiments that is recorded on the digital fashion, basically, not on the conventional fashion. That definitely allows you to optimize the way by which you do experiments and, of course, reduce costs and time respectively. And the third part is related to the manufacturing industry, manufacturing part of the pharma and biotech, where any batch failures that happens in the manufacturing process can be investigated thereupon. And the time of investigation is very, very critical in the industry. So a manufacturing analytics program identifies, finds the cause of a particular investigation that is that happens through the basically through the what I say quality management system. As Mark was mentioning in his presentation related to sticking in tablet, you come out with a way using Material Studio to identify certain specific aspects, property related to sticking. But if in the batch a sticking in tablet has been identified in the quality department, how do you find the root cause of that particular? Although Mark has said that it is adhesion, moisture, and other properties, how do we relate to this and how do we monitor the whole process is what process manufacturing analytics is all about. And Biovia has a very big strength on it with 22 plus years of experience and 180 installations across the world. And following that, Biovia also has a set of tools related to the document management system and quality management system, which is compliant as per the guidelines that is defined by the FDA, specifically 21 CFR Part 11 compliance and other compliances. And of course, we have also acquired a company called Medidata, and Medidata is one of the biggest organizations related to clinical trials, and of course all your artificial intelligence, machine learning is all part of Medidata's offering along with Biovia. So this is the whole portfolio, and you see the whole cycle from the ideation up to the clinical trials. The whole product is on a single platform that we offer. That's the greatness of this particular Biovia tools. Thank you.
Great, sir. Thank you so much for giving such a wonderful explanation. And we have few questions to Dr. Mark also. Let me read that question. There is a question from a chat. How we can test different materials before packing with API? Can we use Material Studio solutions or any Biovia solutions for this?
Can we test different materials before packing with API? So yes. The whole idea of running those simulations is to do some in silico testing. So we model the materials and we can have various formulations. So we can have the API with an excipient or another excipient or the API in the solvent. And then for each one of those combinations, we can calculate the properties, the relevant properties, such as a crystal structure. I think that was a question. Oh, yes.
Yes, okay. Thank you so much, sir. And there are a few questions from the chat. I would like to pick a very important thing. And there are a few questions for what are the Biovia solution and tools will accelerate these results? I mean, the pharmaceutical companies' results. That's
Okay, this is for me.
yes,
Biovia, sorry.
Yes,
Okay.
it is given as general actually. What are the Biovia solutions and tools will accelerate these results? That means pharmaceutical results. That's what I mean.
Okay, fine. As I mentioned initially, the set of collaborative tools that we offer, and one of the tools is called as Discovery Studio. That's the life science modeling and simulation. And as Mark was explaining about Material Studio, that's one of the exquisite tools that we have related to pharmaceutical industry. And Mark has a very wide experience working on that, and he's a fellow on that, fellow with respect to Material Studio research, and one of the distinguished researchers basically. And in addition to that, we have certain tools related to artificial intelligence and machine learning for the innovation part, and that is attributed to a product called as Pipeline Pilot that has different collections, from the chemistry collections to biology collections to instrumentation collections to even a clinical trials basically on the Thomson Reuters, which is called as Quarterless collection now, and those collections are all available within the program. And not only that, any research that is attributed to a researcher will be totally documented through an electronic lab notebook system, and a discovery of a small molecule or a macromolecule will be attributed through a chemical registration system and a biological registration system. So these are a partial list of tools and applications that Biovia has to offer for research and innovation cycle. And Mark, if you want to add anything, please feel free.
No, it's good description, Alan.
Thank you.
All right. There is also one more question from the participants. Generative therapeutics is a software and have a good solution in pharmaceutical industry.
Yes, generative therapeutics is, I would not say that as a software, it is an experience, that's what we define it, and it's powered by what is called the 3D experience. And Dassault System is one of the renowned companies that offer what is called as a 3D experience platform that is spread across multiple industry segments, 11 different industry segments, starting from automotive to consumer packaged goods to construction to oil and gas to chemicals. And with their being in the pharmaceutical industry, generative therapeutics design is one of the greatest solutions that they offer on the platform, which is specifically collaborative, innovative, and also has a capability to share and deploy the results. So that's the terming it as a software would be more general. I would say that as it's a 3D experience platform for drug discovery and innovation. That's the right term I would use.
I hope participants got from this. This is not a software. This is 3D experience, a solution from Biovia. And there is also one question. How the Material Studio has an application in tablet manufacturing?
So yeah, I would invite anybody interested in the particular topic to go to the Biovia website, where at the end of this week, the various the recorded presentation can be found. I think it'll be released on Friday. We gave the presentation just a week ago, and I gave a presentation on this tablating, and together with a colleague from Simulia, which is another Dassault System brand, running larger scale simulation. Abacus is known for finite element analysis. Okay, so in the case of tablating, what we use Material Studio for is to predict the crystal size and shape using the morphology tool. We try to estimate the molecular interaction between the API in the excipients with the punch surface, the stainless steel surface of the punch. And so that will give us an indication of the likelihood for stickiness. And we could, because we are in silico, we could easily screen different formulation, that is the type of excipient, the concentration, and so forth, in order to minimize the stickiness that we're trying to avoid.
Thank you so much for giving a wonderful explanation. And we have one more question. Can Discovery Studio can be utilized for screening bioactive natural products?
Yes, a wonderful question, because people are trying to go natural now, and more from the synthesis part in terms of bringing down the toxicity basically. So it somehow correlates to my panel discussion question that you actually put forth. Yes, Discovery Studio has a capability to screen natural compounds. The screening can be done in multiple ways. It could be function-based screening, basically. Certain functional aspect of the chemistry of the molecule can be taken. It could be based on the similarity coefficient that you can basically screen it. You can also screen it based on different pharmacophore features, basically existence of a hydrogen bond donor or an acceptor, which readily actually interacts with certain specific constituents in the protein target. That's one way in which you can also screen it. In addition to that, I would also suggest that docking is one of the initial programs that people start using into screen compounds. So natural compounds very well sits within this portfolio of acceptance by Discovery Studio for docking against these protein targets. So yes, it can be done. We have a very good program called as a ligand profiler, which hosts the largest pharmacophore database of protein structures and targets in the program. And any natural compound that you have, if you want to know what is the target associated with that, you can shoot your small molecule against these pharmacophores basically, and it will screen across all of them and give you a fit score which will identify what could be the probable target for your natural compounds. And that's one of the best ways that which you will identify your target too. So yes, these are the different methods. Of course, there are multiple ways by which you can do it. We can definitely get into the details separately also, because it will take a lot of time. Thank you.
And there is one general question from the audience. Will Biovia conduct internships on computational chemistry?
Sorry, what is the question?
Yeah, can Biovia will conduct an internships or workshops on computational chemistry?
Yes, Biovia has a very active partnership network, and Altum being on the top. And Altum has definitely helped us a lot in working with many of our customers and bringing in a lot of enthusiastic and active researchers using our applications. And Dassault Systems Biovia, along with Altum, will definitely be one of the key providers for these types of internships in computational chemistry and biology. Yes, going forward, yes, we can.
Thank you, sir. Thank you so much. And I believe that both Material Studio and Discovery Studio, Generative Therapeutics, and Biovia has end-to-end solutions for computational chemistry and biology in pharmaceutical therapeutics. And I hope the people are watching this video and pharmaceutical people basically will get benefit out of this. And with this, I thank all the people and I hand over the session to Dr. Tanushree.
Okay, so we have come to the end of the session now. And thank you, Mrs. Divya. And I would also like to express my gratitude to both the eminent speakers, Dr. Anand Krishnamurthy and Dr. Yune, for taking out time from their busy schedule and enlightening us. It's a lot of takeaway learning experience. Thank you, sir. Also, I would like to thank all the participants for attending this session. In case of if any participant has any product query, so kindly reach out the number that is being displayed on the screen, and also you can email. So email ID is also displayed on the screen. And don't forget to fill the feedback form. Feedback link will be shared in the chat box. Kindly share your views on this screen. Thank you all and take care.
Thank you.
Thank you very much.
Thank you.
Thank you very much. Bye now.
Yeah, thanks, Dr. Tanushree.